Two people sit down to the same dessert. One finds it almost too sweet and stops after a few bites. The other barely registers the sweetness and keeps going. Same sugar, two completely different experiences, and over years, two different bodies. Part of the reason is written in their genes, but there's a twist. The carbohydrates themselves write back, switching genes on and off. This is the two-way conversation between your DNA and the carbs you eat. Carbohydrates are the body's largest source of energy, 45 to 65% of your daily calories, and the science splits cleanly in two. First, nutrigenetics, how your genes shape the way you taste, crave, absorb, and burn carbs. Second, nutrigenomics, how those same carbs reshape the activity of your genes. Start with taste. Sweetness isn't fixed, it's a genetic sense. Two receptors, built from a pair of sweet taste genes, sit on your tongue and detect sweet molecules, and small variations tune them. A single variant explained about 16% of the differences between people in how sweet they find sugar. Another was linked to an 18% drop in how much sugar people ate. The pattern is consistent. The less sweetness you perceive, the more sugar you tend to eat to reach the same satisfaction. Taste is only the start. Sugar is also rewarding, and reward runs through the brain's dopamine system. A variant in the dopamine receptor gene has been tied to eating less sucrose. That same receptor sits at the center of food reward and even food addiction. Appetite itself is partly inherited too. A variant in one appetite gene shifted people toward 2.6% more carbohydrate and away from fat. And a variant in another, the TUB gene, tracked with higher sugar intake and a higher glycemic load. Different genes, same theme. How much you want carbs is written, in part, in your DNA. Some of the strongest effects come from a sugar sensor. GLUT2 is a glucose transporter that also acts as a detector, and deep in the hypothalamus, the brain's appetite center, it gauges how much sugar is on board. One variant was linked to a striking 30% increase in carbohydrate consumption, independent of protein, fat, alcohol, or age. The leading idea? The sensor slightly misreads the body's glucose, so the brain keeps asking for more. The same variant has been tied to higher rates of type 2 diabetes. And a genome-wide study of 1,700 women found another, a risk variant that added about 2.5 grams of carbohydrate a day for every copy. Like GLUT2, it acts in the hypothalamus. Now flip the conversation. The carbohydrates you eat don't just feed you, they change which genes are switched on. Fructose is the clearest example. Fed in excess, it reprograms the liver, ramping up the genes that turn sugar into fat while turning down the liver's antioxidant defenses. The result? More triglycerides, more stored fat, more oxidative stress. Glucose does its own rewriting. Together with insulin, it switches on the machinery of fat synthesis. The food isn't passive, it's an instruction. And this rewriting can reach across generations. In animal studies, a mother's high fructose diet during pregnancy altered her offspring's gene expression and programmed lasting changes, including higher blood pressure later in life. It's an early, sobering hint that the carbohydrate environment in the womb can leave a genetic signature on the next generation. Some of this conversation is ancient. Take starch, the carbohydrate in bread, rice, and potatoes. It's broken down by salivary amylase, made by a single gene. And populations with long histories of starch-rich diets tend to carry more copies of that gene, the genome adapting to the menu over generations. And not all starch behaves the same. Resistant starch escapes digestion and feeds your gut bacteria, which return the favor, producing short-chain fatty acids and, in studies, lowering circulating cholesterol. Put it together, and you get what researchers call your nutritional phenotype, the unique way your genes and your diet shape each other. It's why a single carbohydrate guideline can't fit everyone. Two people can taste sugar differently, crave it differently, absorb it differently, and be reprogrammed by it differently. Much of the deepest evidence still comes from animal models, and the field is young, but the direction is clear. Carbohydrates are not just calories. They are a conversation, your genes shaping how you handle them, and the carbs themselves editing your genes in return. So here's the question for the future of nutrition. Once we can read both sides of that conversation, what belongs on your plate?